Hi, my name is Callie Chappelle, and welcome to this video about ribosomal subunit exchange. This is made for MCDB 427, which is molecular biology at the University of Michigan. So before we get started, I just want to remind you about ribosomes. So ribosomes in prokaryotes are 70S, and that's the monosome. So we call the ribosome that contains both the large and small subunit a monosome. And as I said, it contains the large subunit, which is 50S, and the small subunit, which is 30S, and this is in prokaryotes. And the big question that we want to answer by this experiment is, do these subunits dissociate after translation, or do they stay together? Are they bound together, or do they come apart and then reassociate? So in order to figure this out, we are going to do a pulse chase experiment. And what I'm going to do in this video is I'm going to quick introduce the experiment to you so you get a kind of general idea of where we're going. I'm going to go in depth on the first experiment they did in order to kind of have a very elaborate control, I guess. And then I'm going to go more in depth into the details of this experiment. So what they do is they're going to do a pulse chase experiment. So remember what a pulse chase experiment is. It's an experiment where first they're going to label the creation of something, in this case ribosomes, and then they're going to chase it by following it with non-radioactive cold things. So they're going to grow it in a cold medium, and then they're going to ask the question, what is the association of the hot things with one another, or with cold things? So the first thing they're going to do is label E. coli ribosomes by growing E. coli in heavy medium and with tritium as a radioactive tracer, so this is the pulse. Then they're going to chase it by growing the cells in light medium, so the ribosomes produced after this will be cold. And then after 3.5 generations, they're going to separate out these ribosomes by using a sucrose gradient, which separates by size, and we're just going to say it by size, and detect the radioactivity. Let's jump into the first experiment they did, which was not this, it was a control. So before we explore what happens in the pulse chase experiment, let's look at what the ribosomes look like when they're grown in heavy and light media separately. So the first thing they do is not a pulse chase, alright? Everything is going to be radiolabeled. So they're going to grow E. coli ribosomes, they're going to grow E. coli, which are producing ribosomes, in heavy media, and then they're going to separately grow the E. coli that are producing ribosomes in light media. So let me tell you about what's in the heavy media. And in the heavy media, we've got 15N, 13C, and 2H, which are all heavy isotopes. And the radioactive one that we're going to be watching that we're going to be labeling with, is going to be tritium. In the light media, it's called light, which is a little bit of a misnomer because they're just going to use regular nitrogen, carbon, and hydrogen. And they're going to label these guys so we can follow them with 14C. So we're going to grow our E. coli, we're going to harvest our ribosomes, and remember, now our ribosomes are respectively labeled with tritium and 14C, and then we're going to mix them together. We're going to mix the heavy and light ribosomes together, which are shown here. And what's in there? So we've got our monosome, which is 86, and then we're going to have our large subunit, which is going to be 61S from the heavy, and our small subunit, which is 38S from the heavy. And the same with the light, we're going to have our monosome, which is 70S, we're going to have our large subunit, which is 50S, and our small subunit, which is 30S. And really the reason why we're doing the controls is because we actually want to figure out these numbers, if this is 86S, this is 61S, this is 38S, etc. So in order to do that, we're going to run on a sucrose gradient, which separates things out by size, where large stuff goes to the bottom and small stuff comes to the top. It'll go in this order, and after we run our sucrose gradient, what we do is, remember, we cut out all those we're going to slice out a bunch of discs, and then measure radioactivity in each. We're going to be slicing out what we call fractions to get this kind of graph. So let me show you what this graph is. On the x-axis is fraction number, where large stuff, which comes from the bottom of the sucrose gradient, is here on the left side, and small stuff, which is stuff on the top of the sucrose gradient, is here on the right side. And the y-axis is radioactivity. So it's tritium, remember, to measure concentration of the things grown in heavy, and it's 14C for things that are grown in the light. And so this is what we see. So the heaviest thing, so the thing that goes to the bottom, is going to be the monosome of the things grown in heavy media, which we find is 86S, right? This is what this tells us. The next heaviest thing is going to be the monosome from things grown in light, which is 70S. That's what this is what this experiment tells us. The next heaviest thing is going to be the large subunit grown in heavy, which is 61s, and then the large subunit grown in light, which is 50s. The small subunit grown in heavy is 38s, and the lightest thing is going to be the small subunit grown in light, which is 30s. So why is this a control? Well, it tells us where the ribosomes that are grown continuously in either heavy, which is, remember, labeled with tritium, or light, which is, not labeled with 12C, but 14C, would migrate on a sucrose gradient, which allows us to compare the migration sizes when we actually end up doing this pulse chase experiment, where we want to ask, does exchange occur, right? So it'll allow us to see whether subunit exchange occurs, because we're going to see a shift in these peaks in one of these directions. Not to ruin the surprise. Let's look at what exactly is going on in the pulse chase experiment. So here's the ribosomal exchange experiment. This is the second one. And remember, the method is the pulse chase. So the first thing we're going to do is label. So remember, pulse chase, first we're going to pulse with something radioactive. We're going to chase with something that's cold. So pulse with hot, chase with cold. What are we pulsing with? We're labeling E. coli ribosomes by growing E. coli and therefore producing ribosomes in heavy medium. Remember, so it's 15N, 13C, and 2H. With tritium as a radioactive tracer, that's the pulse. All right, we're just adding a very small amount of radioactive tracer. So in this experiment, remember, what we want to know is, do the ribosomal subunits exchange? And so so we have two different things that could happen. The first is there could be no subunit exchange, which I'm going to draw in this area. And then if subunit exchange does happen, which I'm going to draw in this area. So for this first step, we're going to just be growing heavy. Then we're going to switch our cells to grow in light media. This is the chase. So remember, this is just normal 14N, 12C, and hydrogen. And this is cold and not radiolabeled. So this differs from the previous experiment, remember, where we grew the light stuff in 14C. This is not labeled. This is cold, all right? And this is where we're going to see a difference if there's subunit exchange or no subunit exchange. If there's no subunit exchange, when we grow stuff in light, the light will just form on their own, right? We're going to get monosomes that are of just light, and then we'll have monosomes of just heavy, and we won't have an intermediate. But if there is a subunit exchange, as we grow in light, we'll get exchange between large and small subunits between cold and hot. So what we'll get is an intermediate set of nucleosomes where we have a large subunit from hot coupled with a small subunit from cold, and we will have and conversely we'll have a large subunit of cold coupled with a small subunit from the hot, and this is going to be what I call an intermediate. Also, we're going to get monosomes of just the cold as well. But remember, ultimately what we're going to be looking at is radioactivity, and because these will be cold, we are actually not going to see them. They'll be invisible. So we're going to grow in light medium. After 3.5 generations, we're gonna collect our ribosomes, and then we're going to add something else. Okay, and this is what I think is like the most confusing part. I almost said confusingest. Wow, the most confusing part of this experiment. And that is completely separately of these guys, we're going to grow some ribosomes in light medium, okay, with 14N, 12C, 1H, but we're going to add 14C, all right? So separately, we've grown this with 14C. We collect our ribosomes, and contained in that fraction are going to be 14C labeled 70, 14C, 70S monosomes, 14C large subunit, 14C small subunit. Remember, this is this step over here that we did, right? And what we're going to do is, before we run it on the sucrose gradient, but after we've collected these guys, we're going to add those in. And all that does is just tell us where the sizes of our products are in reference to these guys, in reference to what we're adding, okay? It just creates kind of like what I like to think of as, like, not like a ladder, but a standard against which we can compare sizes. All right, so keep in mind there still are subunits that we're growing that the exchange would have happened with that are cold, but now we just have this standard, if you will, that we can compare what happened in the actual experiment. Right, in the actual part where we're testing different things. So we're going to add these 14C labeled lights in. So then we're going to separate using our sucrose gradient, and then we're going to detect radioactivity. So here are the sucrose gradients that we would expect in these two different scenarios. So imagine that there's no exchange happening at all. What would we expect to see just from this part of the experiment, not even thinking about the addition of the light? Well, we'd expect to see heavy we'd expect to see the heavy monosome, we'd expect to see just the heavy large subunit and just the heavy small subunit. And we wouldn't see any light from this because the light's not radioactive. But we're going to add light back in, right, just as our size standards, just think about it as our size standards, and so we'll see the light monosome, the light large subunit, and the light small subunit as well. All right, so these things that are in purple these things shown in purple are the things that were actually created, in the pulse chase, right? And then these things that are shown here in blue, that's the added light, the radioactive light that we did as our size standards. Now, what would happen if we had an exchange? Well, we're still going to see the same radioactive light that we just added because we added it as a standard. And we should see the heavy shouldn't change, right? We still are going to see large and small subunits alone that were created in the heavy that are just not associated with another subunit. But instead of having just the large subunit, right, that's always associated, that the large and small subunit associated that are both heavy, we're going to get this intermediate thing, remember? We're going to see exchange between the cold light and the hot heavy to create an intermediate thing. And because it's intermediate with things that were grown in light and heavy, it's going to have a different mass than the light or the heavy alone, the light and heavy monosomes alone. And we're going to get this intermediate, which is 78S. And how do we figure that out? By looking at where it falls in sucrose gradient. Let's look at the results. But before I give you the results, just attempt to, let's make a prediction about what we think we would see, all right? Let's make a prediction about what we think we would see. Well, first, I would predict that because we're talking about exchange, that there shouldn't be any major changes in the large and small subunits themselves. So I wouldn't expect them to degrade or to break down or get larger for any reason. So I would expect to see the large and small subunits to be the same. Next, what would I expect to see about the light? Well, because we're adding the light subunit, the light labeled ones in later, I wouldn't expect to see any differences between the light. I would expect to see, so this is just the size control, this is what I just showed you over here. I would expect those to be in the exact same places because essentially do the exact same thing and just add it later. So light should be the same. And finally, I would expect to see the 86S to disappear. 86S should disappear. Now, take my word for it right now, but I'm going to explain exactly why in a second after I go through the rest of this, all right? So we're going to come back to this guy. So here are the results. Ignore this green thing really quick. Everything else is what we actually saw. So, like we expected, the light is exactly the same. Our 30S is in the same place, our 50S is in the same place, our 70S is in the same place. And remember, the reason why that is because we don't add the light labeled stuff until after the experiment's already done. We would expect to see the large and small subunits the same, and indeed we do. So here are the large and small subunits. Remember, here are the large, and here are the small. We see exactly the same, 30S, 38S for small, 30, 38S, 50, 61, 50, 61 for the large. All right, does the 86S disappear? Yes, it does. I just kind of drew it in here just so you'd know where it would be if it was there, but it's not. It's just like a ghost. It even kind of looks like a ghost. Ooh. Anyway. Sorry, I cracked myself up. And what we do see, though, is 70S in the same place, right? That was shown up here. But we see this weird 78S bump. So what could that be? Well, remember, what we're wondering is, do the subunits exchange? And if they exchange, it produces this intermediate thing that's an intermediate size, right? Because we've got one grown in light and one grown in heavy. And that was shown here. This is the 78S. And indeed, we see the 78S here. This is evidence that shows that the subunits do exchange because we see an intermediate that is of intermediate size. So what are the results? The first thing is that the 78S monosome, sorry, spelling is terrible, monosome formed by exchanged particles. And the second thing that we see is that the subunits are stable. Subunits stable. This is what I'm talking about here, where the large and small subunits should be the same. Essentially, we don't see any pig shifts between the large and small, and that indicates that they aren't coming apart and reforming. There's no reduction in size. All that's exchanging are between subunits, not within the subunits themselves. Before I move on, I just want to show a quick diagram of what each of these peaks represent, like I did over here, like this. I feel like that helps me at least understand this. So the 78 peak represents either the large subunit or the small subunit that's heavy mixed with a light unlabeled subunit. The 70S is, remember what we added, so that's the monosome for the light and labeled. 61S is the large subunit that was grown in heavy, that's labeled. The 50S is the large subunit grown in light, that's also labeled, that was what we added later. The 38S is the small subunit that's labeled. And the 30S is the small subunit that is labeled that's grown in light, that we added later. Alright, so the ones that we added later were this, this, and this. Oh, sorry, I switched blues. That doesn't mean anything. Added later. Remember, these were the labeled 14C. Keep in mind that these small subunits, these light subunits are unlabeled. The reason why we can see them is because they are paired with a tritiated heavy, tritiated subunits that were grown in heavy, and these two are encompassed by the 78S peak. The difference, yes, I know that the heavy is the large subunit here and the small subunit here. The difference in size is so minuscule that it just shows up as one peak. The last thing I want to go over is back when we were talking about this. So one thing that I mentioned was when we get this intermediate, the production of this intermediate after 3.5 generations, we don't see reassociation of two heavy labeled guys together. We don't see association of a heavy labeled large and a heavy labeled small. And I'm going to go through why that is right now. And in order to do this, we're going to take a little closer look at what's going on during the chase. So during the pulse, everything gets labeled, right? All the ribosomes get labeled as heavy. But when we switch over to our unlabeled light media during the chase, now all subsequent ribosomes that are formed, or ribosomal subunits that are formed, are now light. And so this really helped me understand what was going on. So we start out with, after the pulse, all heavy. But after one generation with a chase, we see a division that happens, right? So the heavy stays heavy. This is the same. That's why I said same here. But it produces one light monosome, right? It produces two subunits, one large, one small, that are light. And this is because it's growing in light, right? And so after 2 generations of the chase, we still have our initial one grown in heavy, but now that one again produces a light, and then the light produces 2 more lights. And after the 3rd generation, again, we have our same heavy one here that produces 1 light. This light one produces 1 light. This light one produces 1 light, and this light one produces one light. So in total, we have, let's say we just start out with 1 ribosome. We have 8-fold more ribosomes. 1 is still heavy, and 8 of them are light. Okay, so let's take a look at what's going on after 1 generation. So the 2 key ones that you should focus in on are how many we have after 1 generation and how many we have after 3 generations. So after 1 generation, we have 1 of each, essentially. We have 1 heavy large, we have 1 small, we have 1 heavy small, we have 1 light large, and we have 1 light small. And so then we want to know, well, what combinations of monosomes could we have? So we could have a large heavy and a small heavy. We could have a large heavy and a small light. We could have a large light and a small heavy. And then we could have a large, oops, that's the wrong color. We could have a large light and a small light. And these are all perfectly possible things that we could have. But remember that the way that we actually see these guys is by looking at radioactivity. So we don't actually see the light on light guy at all. It's invisible. So when we want to think about the probability of getting any of these and for them to show up on our gradient, we're only looking at the population of ones that are already labeled. So within this, 50% chance that we'll get, 2 out of 3% chance that we're going to get an intermediate, and a 1 out of 3 chance we're going to get just the heavy labeled. All right. But now, I want to explain this conceptually, and then I'm going to show you just a little bit of math, don't get freaked out. But now, as we have more and more generations, we're getting more and more light. So the probability that one of these heavy subunits associates with light is getting greater and greater and greater. There's more cold subunits, so it's more likely to have a hot-cold combination than a hot-hot combination, all right? And when we have a hot-cold combination, remember, that makes it intermediate. It makes it more likely for it to be 78S. So after 3 generations, or after 3.5 generations, pretty much all of our monosomes that are labeled, right, are going to be intermediate simply because of probability. Let me show you what this means in terms of a table. So if I number all of these guys, and 1 represents the heavies, and 2 through 8 are all light, what are all the possible combinations that we can make? So 1 on 1 is going to be what? 86S. 1 on 1 is going to be an 86S. Oops, these numbers just got off. Weird, sorry, let me just renumber this for you right now. This is 1, this is 2, 3, 4, 5, 6, 7, 8, I didn't notice that. So 1 paired with 1 is going to be an 86S, 1 paired with 2, so that would be like this paired with this is going to be a 78S, 1 paired with 3 is going to be 78S, 1 paired with 4 is going to be 78S, and so on. But remember, if 2 was paired with 3, for example, if 2 was paired with 3, this is not going to be radiolabeled because it doesn't include subunits, either of 1 subunits, right? So all of these things that say ND, these are not visible, okay? So we can go through and calculate the probability that we have 78S over an 86S, and you can just see there's way more lilac here than there is dark purple, which indicates that pretty much all after 3.5 generations, or after, in this case, 3 generations, are going to be intermediate, which is why we see just the 78S. But let's take a look at what this graph would look like when we have just after 1 generation. So remember, after 1 generation, 2/3 of them are intermediate, but a whole 1/3 is not. A whole 1/3 is heavy on heavy. So if I wanted to show you what that data would look like, there'd be a peak here after 8. There would be a small peak here at 86, and you'd see our other peak at 78, where these intermediates represent the one at 78, and where this heavy on heavy represents the small one at 86. Okay, and this would be approximately 1/3 the size of this. This should be 2/3 because of the statistics I just showed you. However, when it's not a whole 1/3 that's heavy on heavy, when it's just this tiny amount, don't quote me on this, I think it's like 6.7, I don't know, I don't remember, don't quote me on that. When it's a much smaller amount that's heavy on heavy, essentially you don't see that little bump here at 86. It's really just flat because that's so minuscule you just don't see it. It's all intermediate, which is why you see just the 78S. I hope this video was helpful. I had a lot of fun making this, and I had some discoveries. And what's pretty cool about this experiment, I think, kind of to wrap this up, is it seems really simple, but it's actually deceptively complicated. So I hope that I went through everything you need to know in this video.